Hello, my name is Pathanasak Lung Siriwanit. I am PhD candidate, Department of Biology, Faculty of Science, Chiang Mai University. I have received the RRI scholarship with grant number PHD60I0089. Today I would like to present in the topic antibacterial activity and tannin tolerance of Bacillus species isolated from leaves of Miang, Camellia sinensis, variety Assamica. Miang or Assam tea has a scientific name as Camellia sinensis, variety Assamica. Miang plants have been found that can grow at an altitude of 1,000 to 2,000 meters above sea level. By local wisdom of northern Thai people, fresh miang leaves have been used as the raw material for fermentation to produce a chilling refreshment product called fermented miang, that is, fermented by lactic acid bacteria. Miang or Assam tea plants are an indigenous plant in highland of northern Thailand, which can be found in Chiang Mai, Chiang Rai, Phra Nath, Lampang, and Mae Hong Son province. However, from past to present, there is less study about the microorganisms on Assam tea leaves and the utilization of these microorganisms. So, this research aims to evaluate some biological properties of Bacillus isolated from miang leaves in order to rule out the role of the microbes on miang fermentation process. Firstly, miang leaves were collected from Chiang Mai and Phra province in northern Thailand, and bacterial isolation was performed within 24 hours. The colony with characteristics of Bacillus species were selected purified prior to determination of their biological activity. 16 bacterial isolates showed the different colony morphology of Bacillus, which are isolated from 10 miang leaves. Based on molecular identification, all bacterial isolates were identified within 3 species including Bacillus thuringiensis, Bacillus siamensis, and Bacillus licheniformis, which had the similarity of 16S RNA gene sequence related to their Thai strain between 99.2 and 99.9%. Bacillus has been reported that can be found in the environment. Bacillus have been found to play a vital role in health and economy. For determination of antimicrobial substance production by an agar-well diffusion method, the results show that the culture broth of Bacillus licheniformis ML075-1 could inhibit growth of Bacillus cereus and Staphylococcus aureus, and Bacillus siamensis ML122-2 could inhibit growth of Staphylococcus aureus and methicillin-resistant Staphylococcus aureus, while all the strain of Bacillus thuringiensis could not inhibit trace pathogenic bacteria. However, antimicrobial activity of Bacillus isolate was decreased after growing in the medium supplemented with calcium carbonate. For determination of tannin tolerance, the results show that only Bacillus siamensis ML122-2 could grow on the medium containing 1% of tannic acid. The bacterium was grown inhibited when concentration of tannic acid was higher. In conclusion, Bacillus found in miang leaves can inhibit growth of some pathogenic bacteria. The narrow or broad spectrum properties depends on species of Bacillus, and interestingly, Bacillus siamensis ML122-2 can inhibit growth of methicillin-resistant Staphylococcus aureus, and it can also tolerate tannic acid. Finally, I would like to thank Assistant Professor Dr. Narumon Thongbai, Teaching Assistant and Research Assistant Scholarship, Graduate School Chiang Mai University, Tea Gallery Group, Amazing Tea Limited Partnership, the Research and Researcher for Industry Scholarship, the National Research Council of Thailand, and Department of Biology, Faculty of Science, Chiang Mai University. This is some reference in this research. Thank you for your kind attention.